Hello friends, welcome to KLS Gokte PU College of Commerce and Science, Belgavi. So this is our fourth lecture on a topic, surface chemistry. This is Mr. Akash Vasan Nesarkar, lecturer in chemistry, Department of Chemistry. So let us see the contents of today's topic. The first one, that is important features of solid catalysis, shape selective catalyst, application of zeolite, enzyme catalysis, and lastly characteristics of enzyme catalysis. So we will be discussing these 5 points in today's class. So before we proceed to the actual contents, it's glad to know the Nobel Prize winners of chemistry of this year. So, the Nobel Prize winners in chemistry 2020 is awarded the two duos, that is Emmanuel Charpentier and Jennifer A. Doudna, a French biochemist and American biochemist. They got the Nobel Prize winner for the development of gene editing tool, and this is a great success in chemistry or human science or human life. By using the gene editing tool, the researchers can change the DNA of plants, animals and microorganisms with the high accuracy. So it is a proud moment of us that something is good has happened in this COVID-19 year. So now let us begin to our actual syllabus. The important features of solid catalysis. So the first one, that is the activity of catalysis. The activity of catalysis is nothing but the ability of the catalyst to increase the rate of a chemical reaction. So catalyst has a certain ability that it will change the path of the reaction by lowering the activation energy. So that is its ability. By using those paths and by lowering the activation energy, it will increase the rate of the reaction. So that is called as an activity of a catalysis. So it is nothing but simply ability of a catalysis to increase the rate of a chemical reaction. The second one, selectivity of a catalyst. This is very important, guys. It is the ability of a catalyst to direct a reaction to yield a particular product, excluding others. So some reactions requires selective catalyst to yield the proper product. Suppose we want to prepare a methane. To prepare methane, there is a selective catalyst. Means a proper catalyst has to be used to get a methane. If we use other catalyst instead of the proper catalyst, then we will get the different product. So that is called as a selectivity. So selective catalyst will give the selective product. Got it? So that is called as a selectivity of catalyst. It is the ability of catalyst to direct a reaction to yield a particular product. So in order to get a particular product, we have to use a particular catalyst. So please make down a note. Action of catalyst is highly specific in nature. That is, given substance can act as a catalyst only in a particular reaction. So that is very important. Some catalyst becomes active only in a particular reaction. So that is called as a selectivity of catalyst. For example, when carbon monoxide and hydrogen react to form different products in the presence of different catalysts as follows. So let us see this various examples or various reactions by using the different catalysts. So when carbon monoxide and hydrogen react each other in the presence of nickel catalyst, the first catalyst we are using, that is a nickel catalyst, then we'll get the methane as a main product and water as a byproduct. So that is called as a methanization. So when carbon monoxide reacts with a hydrogen in the presence of nickel catalyst, then it gives a methane as a major product. But instead of nickel, if we use or if we change the catalyst, that is copper, chromium and zinc oxide. Instead of nickel catalyst, if we use the copper, chromium and zinc oxide as a catalyst, then we'll get methanol as our major product. So that is called as a selectivity. So we get the product based on the catalyst, which catalyst we are using. The reactants are remaining same. We are having carbon monoxide and hydrogen to our left hand side, but the product, which product we want, it depends upon the catalyst. If you use the nickel catalyst, then we'll get methane. If you use the copper, chromium or zinc oxide, then we'll get methanol. So that is what the selectivity of catalyst. So we'll get the product based on the selectivity of catalyst. If we use nickel, then we'll get methane. If we use zinc oxide, then we'll get methanol. Getting? The next one, instead of zinc oxide, if we use the catalyst as a iron or cobalt. So when carbon monoxide reacts with the hydrogen in the presence of iron and cobalt as a catalyst, then we'll get Fischer-Tropsch synthesis. That is, we'll get the hydrocarbon and water as a byproduct. So if we use iron and cobalt as a catalyst, then we'll get a product that is a hydrocarbon. So instead of iron and cobalt, if we use the another catalyst, so when carbon monoxide reacts with the hydrogen in the presence of rhodium cluster catalyst, then we'll get glycol, that is union carbide. We'll get glycol as a major product. So this is what the selectivity of catalyst. Our catalyst will select the product. Got it? If we use nickel, we'll get methane. If we use iron or cobalt, then we'll get hydrocarbon. If we use zinc oxide, then we'll get methanol. So focus on the left hand side, that is reactant hand side. The reactants are remaining same, right? The reactants are what? Carbon monoxide and hydrogen. But the catalyst goes on changing. So based on the catalyst, we are getting the various product to our right hand side. So that is called as a selectivity of catalyst. Got it? The next one, when ethyl alcohol, that is C2H5OH, undergo acid catalyst, that is dehydration, in the presence of acid catalyst. So when ethyl alcohol undergo dehydration in the presence of acid catalyst, then we'll get ethene and water as a byproduct. So that is also a selectivity of catalyst. Here we are using acid as a catalyst. For example, H2SO4 you can use as a catalyst. Okay. Next one, instead of acid catalyst, if we use the metal catalyst, then instead of hydrocarbon, we'll get dehydrogenation reaction. Instead of dehydration, we'll get dehydrogenation reaction. So let me give the differences between the dehydration and dehydrogenation. So what is the difference between the dehydration and dehydrogenation? So you focus on the byproduct. So on the reactant side, we are having C2H5OH, that is ethyl alcohol. But when ethyl alcohol undergoes reaction in the presence of acid catalyst, it gives C2H4, that is ethene and water. So it removing water molecule, that is called as a dehydration. So removal of water molecule from the alcohol is called as a dehydration, and this reaction is called dehydration reaction, and this take place in the presence of which catalyst? Acid catalyst. You can use sulfuric acid. But instead of acid catalyst, if you use the metal catalyst, then same alcohol it will undergo dehydrogenation. That is, dehydrogenation is nothing but what? Removal of hydrogen. So if we use the acid catalyst, it will undergo dehydration reaction, that is removal of water molecule, and if we use metal catalyst, then it will undergo dehydrogenation reaction, that is what? Removal of hydrogen. So remember guys, dehydration is nothing but removal of water molecule, and this reaction take place in the presence of acid catalyst, and dehydrogenation, that is removal of hydrogen, and this reaction take place in the presence of metal catalyst. So here also we can say that the product is depends on the selectivity of catalyst. Have you understood? Yes. The next one is shape selective catalysis. It is a catalysis which depends upon pore structure of the catalyst and molecular size of the reactant and the product molecules. So this is very important, students. So some catalysts are having different structure. So like zeolite is one of the catalyst which is known as a shape selective catalyst due to their honeycomb structure. So you can see here the structure of ZSM5. It is having the honeycomb like structure, means it is having the pores. See here, pores are present. Pores are nothing but empty spaces. Okay, so that is a particular structure of a catalyst, and in this catalyst the particular small size reactants gets fit into this catalyst and reaction carried out. Are you getting? So zeolites are the examples of shape selective catalyst which is having the honeycomb structure. Okay, let us read the note. Zeolite are microporous aluminosilicate. Zeolites are what? Microporous aluminosilicate with the three-dimensional network silicate in which some silicon atoms are replaced by aluminum atoms, giving aluminum oxygen silicon framework. So in zeolites we are having aluminum oxygen silicon framework, and the structure is a three-dimensional in a such a way that we are having the microporous aluminosilicates. We are having what? Microporous. So pores will be present in the catalyst. Pores are nothing but what? Empty spaces are present in the catalyst. So it is having the aluminum oxygen silicon framework. So zeolites are called as a shape selective catalyst due to their honeycomb structure. But actually what is zeolite? It is a microporous aluminosilicate. It is microporous aluminosilicate with a three-dimensional network silicate which consists of aluminum oxygen silicon framework. So this is one important thing you have to remember, shape selective catalyst. So zeolite is a shape selective catalyst. The next one, let us see the uses of zeolite. So how we can use this zeolite? First one, the zeolite is used as a catalyst in a petrochemical industries for cracking of hydrocarbons and isomerization. So it can be used for the cracking of hydrocarbon into hydrocarbons, and it is also used for the conversion, for example isomerization. So catalyst used as ZSM5. So ZSM5 is a shape selective catalyst. The second application of zeolites is conversion process like xylene isomerization. So zeolites are also used to convert various organic products like xylene, that is called as a isomerization. Also the toluene disproportionation, zeolites can be used for disproportionation reaction. And fourth one, ethyl benzene synthesis and para ethyl toluene synthesis. So zeolites can also be used for the synthesis purposes. So these are the four uses of zeolite. Okay, next, in a human body there are various biochemical reaction take place in a day-to-day life, right? And those biochemical reactions are catalyzed by the catalyst, and those catalysts are called as a biological catalyst, and those biological catalyst is known as enzymes. So enzymes are biological catalysts Enzymes are what? Biological catalysts which enhance the rate of the reaction of biochemical reaction. जो भी हमारे शरीर में रिएक्शन होती है उसको हम क्या कहते हैं? बायोकेमिकल रिएक्शन. और बायोकेमिकल रिएक्शन को भी कैटलिस्ट की ज़रूरत रहती है. सो विच कैटलिस्ट? बायोलॉजिकल कैटलिस्ट. Okay, the certain reaction take place in our human body. Those reactions are called biochemical reaction and the catalysts are called biological catalyst. And उस biological catalyst को हम क्या कहते हैं? enzyme कहते हैं. उस biological catalyst को हम क्या कहते हैं? enzymes. And those enzymes are nothing but complex nitrogenous organic compounds. Those enzymes are nothing but what? Complex nitrogenous organic compounds which are produced by living plants and animals. Which are produced by living plants and animals. So enzymes are biological catalysts which control the biochemical reaction. Right? And these are complex nitrogenous organic compounds which are produced by living plants and animals. Suppose if we had a meal, okay, to digest that meal without the enzyme means what? Without biological catalyst, it takes many years. It takes what? Many years to digest. Got it? So without enzymes, the food cannot be digested. Without enzymes, the food cannot be digested, and therefore in our body there are several biological catalysts are present, and those are called as enzymes. So several enzymes are present to run the biochemical reaction. समझ में आ रहा है? So without enzymes our food cannot be digested. So it is very important, guys. So enzymes are what? Complex nitrogenous organic compounds. Enzymes are what? Complex nitrogenous organic compound, and these are produced by plants and animals. They are actually protein molecules of high molecular mass. They are actually what? Protein molecules of high molecular mass, and they are biochemical catalysts. They are also called as a biochemical catalyst or biological catalyst. Every cell reaction is catalyzed by its own specific enzyme. Like how we discussed earlier, the catalysts are selective in nature. In the same manner, the enzymes are also specific. The enzymes are also what? Specific in nature. For example, inversion of cane sugar. Inversion of cane sugar. To invert, the invertase enzyme converts cane sugar into glucose and fructose. See, this is an example of enzyme catalysis. So, when cane sugar reacts with the water, it undergoes reaction to yield glucose and fructose. Okay, and this reaction is very slow, but when you introduce the enzyme that is invertase, the reaction becomes very fast. Reaction becomes what? Very fast, and therefore the enzyme name is what? Invertase. You know why it is an invert? Okay, because cane sugar will direct the plane polarized light to the clockwise direction, but when it gives the product glucose and fructose, they direct the plane polarized light to the anti-clockwise direction, and therefore it is called as the inversion of cane sugar. So direction of the plane polarized light, it is changed in a polarimeter for this reaction. So we are having the invertase enzyme for the cane sugar. Cane sugar is nothing but what? C12H22O11. The second example is conversion of glucose into ethyl alcohol. The zymase enzyme converts glucose into ethyl alcohol and carbon dioxide. So glucose, the formula is C6H12O6. The glucose can be converted into ethyl alcohol and carbon dioxide. The reaction is very slow, but if we use the zymase, the rate of the reaction, it is increases. So the enzyme which converts glucose to alcohol, that is zymase enzyme. So in examination, they may ask the question for 1 mark like this: which enzyme converts glucose into ethyl alcohol and carbon dioxide? So your answer should be the zymase enzyme. The answer should be what? Zymase enzyme converts glucose into ethyl alcohol and carbon dioxide. The next example, conversion of starch into maltose. The diastase enzyme converts starch into maltose. So we are using here which enzyme? The diastase enzyme which convert starch into maltose. The last example is what? Conversion of maltose into glucose. So to convert the maltose into glucose, the enzyme used is what? Maltase. The enzyme used is what? Maltase. So sometimes the name of the enzyme is derived from the name of the reactant. So here we are having the reactant as a maltose. Therefore we are naming the enzyme as a maltase. So maltose having the formula C12H22O11 and it is converted into 2 moles of glucose. 2 moles of what? Glucose by using the enzyme maltase. So sometimes the name of the enzyme it is derived from the name of the reactant. That also you have to remember. Next, mechanism of enzyme catalysis. So how the mechanism take place? How the mechanism will take place? So there are 2 steps. The first one is what? Binding of enzyme to substrate to form an activated complex. So this is the E stand for enzyme, S stand for substrate. Substrates are nothing but your reactant. So first enzyme binds to substrate to form activated complex which is having high energy. The later on, if the activated complex is having the high energy, means it is unstable. So when the intermediate compound, that is activated complex, is unstable, then it will try to gain the stability upon decomposition. So these activated compounds undergo decomposition to give appropriate product. So the second step is what? Decomposition of the activated complex to form a product. So here activated complex undergo decomposition and it gives product and the enzyme remains as it is. The E remains as it is. We know the function of the catalyst very well. It will increase the rate of the reaction or it will decrease the rate of the reaction, but it do not take the part in the chemical reaction. It will remain as it is after the completion of the reaction. So that is the function of the catalyst. So let us see how the actual mechanism take place via diagram. Let us see. So the mechanism take place in 2 ways. The first one is what? Lock and key model. So लॉक एंड की मॉडल बहुत ही आसान है. यहाँ पे क्या होता है? एंजाइम प्रॉपर शेप रहता है. ओके, एंड हियर यू कैन सी एंजाइम विच विल एक्ट एज अ कैटलिस्ट. सो यहाँ पे एंजाइम का एक प्रॉपर शेप है विच इज हैविंग द एक्टिव साइट. सो दीज आर व्हाट? एक्टिव साइट. लाइक की. लॉक और की मालूम है आपको. सो की कैसी रहती है? सो की इज हैविंग द एक्टिव साइट. इन द सिमिलर मैनर द एंजाइम्स आर हैविंग व्हाट? दिस इज व्हाट? द एक्टिव साइट. Enzyme is having what? Active site. And your substrate, the substrate is nothing but what? The reactant. And the reactant is also having some pores, that is empty spaces. Pores means what? A proper shape you can say. So substrates are also having the active sites. Substrates, on the surface of the substrate it is having the active site. So the substrate fix into the enzyme. जैसे आप की को लॉक में फंसाते हैं उसी तरह से सबस्ट्रेट एंजाइम में फंस जाता है एंड इट फॉर्म एंजाइम सबस्ट्रेट कॉम्प्लेक्स. दिस फॉर्म व्हाट? एंजाइम सबस्ट्रेट कॉम्प्लेक्स विच इज हैविंग हाई एक्टिवेशन एनर्जी. एक्टिवेशन एनर्जी क्या होती है? ज़्यादा रहती है. सो इट इज अनस्टेबल एंड देयर टू अनस्टेबल इट विल अंडरगो डीकम्पोजिशन. सो एंजाइम विल रिमेन्स एज इट इज after the completion of the reaction, but the substrate gets changed into product. So we will get 2 different product. So this mechanism is called as a lock and key model. Okay, आप इमेजिन करो आपके पास लॉक है और की है. की क्या है? एंजाइम है. और लॉक क्या है? सबस्ट्रेट. सो इट इज हैविंग द एक्टिव साइट. बोथ आर हैविंग द एक्टिव साइट. सो दे कैन फिक्स टू ईच अदर एंड इट विल फॉर्म एंजाइम सबस्ट्रेट कॉम्प्लेक्स. And that enzyme substrate complex is having the high energy, so it will undergo decomposition to gain the stability and it form the proper product. And this enzyme remains as it is after the completion of the reaction. after the completion of the reaction. So enzyme को कुछ भी नहीं होता है, enzyme जैसे रहता है before reaction, it will remain as it is after the reaction. Okay, reaction के पहले जैसे रहता है enzyme, वैसे ही reaction के बाद में ही रहता है, ना बल्कि ना उसमें कोई change आता है, nothing. It will support the reaction, that's it. So like this the mechanism take place, and it is called as a lock and key model. Remember, if you remember lock and key model, you can write down very easily, you can write down the answer very easily. So lock and key model. So enzyme mechanism take place in this manner. So some important examples of enzyme catalyzed reaction. Okay, so various reactions are present here. That is, the first one, the conversion of sucrose to glucose and fructose. The source is yeast, it is present in yeast, the reaction takes place in yeast, and the enzyme we are using, that is invertase. The second reaction was glucose to ethyl plus alcohol, that is a zymase enzyme, we are using the zymase, we have seen that, alcohol. The third one, maltose to glucose, it is also present in yeast, and the enzyme name is what? Maltase. The fourth one is diastase, which convert the starch and maltose, the source is malt. The fifth one, amylase, the source is saliva, it converts starch into glucose, it converts what? Starch into glucose. The sixth one, ptyalin, the source is saliva, the enzymatic reaction is it converts the starch to sugar. And the seventh one, urease, this one is important, the urease, it is present in soya bean, it converts urea to ammonia and carbon dioxide. So urease enzyme converts what? Urea into ammonia and carbon dioxide. And last one, pepsin, which is present in stomach, it converts proteins into amino acid. So pepsin is an enzyme which helps to convert protein into amino acid. The last point of today's class, that is the characteristics of enzyme catalysis. The first one, enzymes are highly specific and each enzyme catalyzes a particular reaction. Yes, already we have discussed this. Enzymes are very specific in nature, right? So urease enzyme can be used to convert urea into ammonia and carbon dioxide, but instead of urea we cannot use the other enzyme like maltase. So maltase enzyme is used for maltose only, invertase enzyme is used for cane sugar only. So enzymes are what? Highly specific in nature. Second one, enzymes become inactive in the presence of electrolyte and on exposure to ultraviolet reaction. So the activity of enzyme is reduced, okay, in the presence of electrolytes and ultraviolet radiation. So you cannot use the electrolytes and ultraviolet radiation to carry out the reaction by using the enzyme, because enzyme becomes inactive in the presence of electrolytes and ultraviolet radiation. Third one, enzymes are highly efficient. So very small quantity of enzyme catalyst can increase the speed of the reaction up to million times as compared to uncatalyzed reaction. So it is highly efficient. A very small amount of enzymes we can use, and this small amount of enzyme can increase the rate of the reaction in a million times. So it is highly efficient. Fourth one, enzyme reactions are highly sensitive to catalytic poisoning. Okay, so these enzymes are highly sensitive to hydrogen cyanide, H2S and carbon disulfide. So we have to handle properly if the reaction contains the hydrogen cyanide, carbon dioxide, sulfide. So we have to handle the enzyme properly, because enzymes are very sensitive towards these chemicals, that is hydrogen cyanide, and may they react with the enzyme and it will give different product also. So we have to handle very carefully when hydrogen cyanide, NH2S and carbon disulfides are present in reactions. Next, the activity of an enzyme is maximum at a particular pH called optimum pH, which is between 5 to 7. We know that the pH of a body is how much? 7.4, right? The pH of human body is how much? 7.4. So the activity of enzyme is maximum between the pH range is how much? 5 to 7. So this range is under the acidic condition only. So activity of enzyme is maximum under acidic condition, you can say that. They are active at optimum temperature 298 to 310 Kelvin. So enzymes are active at this temperature only. Means enzymes will show their ability, or it gets active to temperature at a 298 to 310 Kelvin temperature. In this range only they become active. Okay, so these are the characteristics of enzyme catalysis. So thank you, student. So if you have any doubt, you can come in a college and you can ask the doubts. Okay.